Good evening, everybody. Welcome to our patient and family educational session. We're absolutely thrilled to have you all with us this evening. My name is Amy Coote, and I am the CEO of Maddie Riewoldt's Vision. So before we get into the formalities of the night, I would like to start by acknowledging the traditional custodians of the land on which we're meeting. So I'm on the lands of the Wurundjeri people, and our wonderful presenting team from Australia, the Australian National University, are on the lands of the Wall people. I'd like to pay my respects to their elders past and present, and also extend that respect to any other Aboriginal or Torres Strait Islanders who are joining us this evening. As many of you do know, Maddie Riewoldt's Vision is the only organisation in Australia dedicated to driving bone marrow failure syndrome research, advocacy, education, and support for patients and their families. We've now funded over our last 10 years $9.7 million across 36 cutting edge research projects and have multidisciplinary collaborations in place with 24 leading research institutes, hospitals, and universities across Australia and internationally. We are the source of support for bone marrow failure syndromes patients and their families across Australia through our tele-support service, peer support program, and throughout these educational programs. We have now, as of today, supported 160 patients and families across Australia, and we do invite any of you who are with us tonight who have not connected to tele-support or our peer support programs, please do reach out to us because we are here for you when you do need us. So our program this evening will introduce the National Therapeutic mRNA Platform, which is a new research initiative supporting the development of future mRNA therapies in Australia. Our guest speakers will share information about why and how mRNA therapies are being explored for the treatment of bone marrow failure syndromes. They will then be joined by Professor David Ritchie and Dr. Lucy Fox, who will offer a valuable clinical perspective on these emerging therapies. We will have a 30-minute Q&A session at the end, so please feel free to share those questions on the chat or let us know what your big burning questions are. So thanks to everyone who did submit questions in advance. Our speakers have done what they can to take those questions into and bring them into their presentations this evening. So I'm now delighted to introduce our first speaker, Professor Eduardo Iras. Professor Eduardo is the chair of the National Therapeutic mRNA Platform and the director of the Centre of Computational Biomedical Sciences at the Australian National University. Please join me in welcoming Eduardo.
Thank you. Thank you for the opportunity to speak. And can you all see my slides?
Yes, that looks great, Eduardo. Thank you.
Okay, thank you. So thanks very much for the introduction. So I'll give a overview of the technology and the platform, and then my colleagues will continue talking about the potential for therapies and bone marrow failure syndromes. So as you mentioned in the, sorry, I'm hearing some sounds. I'm not sure.
It's just people joining the meeting, Eduardo. Sorry about that.
Okay, it's quite
doorbell.
okay. I'm not sure what's happening. Sounding bells. So as I mentioned in the introduction, so we acknowledge the first Australians and traditional owners of the country where we speak from, the Ngunnawal and Ngari people in the ACT region, and pay our respects to the elders past, present, and emerging. I'd like to start by talking very generally what is mRNA and an overview of this new platform that we are developing. So mRNA stands for messenger RNA, and it can be thought of indeed as a messenger, a molecule in all our cells that carry the instructions of our genomes. And from these instructions, it's a recipe to make proteins, which are the molecules that carry all the functions in in our cells. So these proteins are essential for cells to grow and divide normally, and for all the normal functions to occur in cells, and especially for repair, normal development, and so on. So the mRNA is, as in this cartoon, is trying to portray a middle messenger between the DNA molecules that carry our genetic instructions and these proteins that are essential for function. So the mRNA is essential for medicine, or it's an emerging opportunity for medicine, because most of the diseases are related to the dysfunction of proteins and the dysfunction of essential processes in our cells. And mRNA as a molecule can be made outside the cell, so it can be manufactured and introduced into cells. And once it's introduced into cells, this new mRNA will be produced by the cell, will be taken up by the cell, and will produce the proteins that are not functioning properly in those cells. So we can tell the cell to make a new protein that will make the cell function properly. And so when there is a protein which is causing disease, or a protein that is missing, we can tell the cells how to make these proteins. Now, so the technology exists already, and this presents a very good opportunity for new therapies, but there's been a challenging for several decades, which is when you introduce RNA molecules into cells, and also with experiments, multiple experiments, people realize that these RNAs are rejected somehow. They are not accepted by cells. So these were very soon known to trigger autoimmune responses from the cells where you put these RNA. So after decades of work, Katalin Karikó and Weissman, they found out how to make this mRNA in a specific way that can be accepted by cells. So the cell will recognize this mRNA as non-foreign and will be able to use it to produce protein. So that opens up whole new series of opportunities. So this technology was ready when COVID-19 struck, and that allowed the very quick development of the COVID-19 vaccines based on this mRNA technology. So at that time, it was very quick to design or to produce this mRNA that was specific to target the virus and make it in a way that could be accepted by cells. And now this same approach and the same logic is being applied to all the treatments. How do you choose how to make this mRNA? Can be quite complex in the sense that there are many different possibilities to make this molecule. To give you an idea, so the genetic code that translates the RNA molecule into proteins can function can read the RNA in many multiple ways. That means you can encode the information to make the protein in many multiple ways. So what we want to do is to make this RNA in the best possible way, meaning that the mRNA is stable. So when we put it into cells, it stays there long enough so that it's translated. It produces lots of proteins, so the therapy is effective, and also that it's safe for the individual. That means that we make sure that the mRNA doesn't have any secondary effects So it's very specific to the disease and the cells that we are trying to treat. So that's basically the main goal of the platform, which is um, a project, an initiative funded by the Australian government through the Medical Research Future Fund initiative. Is based on the technologies and the knowledge, the research that we have developed at ANU, and we are now trying to develop it into specific therapies. As a way of summary, so the platform will integrate technologies, computational technologies with experimentation to design these RNAs, to make the RNAs which are stable, optimal for the treatment, specific treatment, and safe for the specific treatments that we are after. We will develop or will implement high throughput testing so that we are sure that the mRNA functions in the way we expect and the way we want and is safe and efficient. And around this, we will generate new technologies, new knowledge, and also opportunities to discuss with regulators and consumer groups to deliver patient-centered, equitable, and impactful therapies. And this is done in a context, a national context, an international context, where we are partnering with companies, with other institutes across Australia, and with academics to make sure that we are able to apply this and put it to good use in new therapies. And with that, I'd like to give the floor to my colleague, Associate Professor Amy George. Thank you very much.
Thank you so much, Professor Iraz. We're really excited about this work and looking forward to hearing from the rest of the team as to how this will potentially apply to bone marrow failure syndrome. So it really does give me great pleasure in inviting our good friend, Associate Professor Amy George, who is the Platform Manager for the National Therapeutic mRNA Platform and lead for the ANU Centre for Therapeutic Discovery. So Amy will be presenting on the application of mRNA therapies for bone marrow failure syndromes. Thanks, Amy.
Thanks, Amy. And thank you, Eduardo. I'm just going to quickly share my slides. Can everybody hear me? Can you see my slides?
Just go to presenter view, Amy, and then we'll be ready to go. Perfect.
Perfect. Wonderful. All right. So yes, thank you, everybody. Thank you for coming tonight. I'm going to be talking tonight about how we might be able to apply these messenger RNA therapies, which Eduardo quite nicely introduced, and how we can actually use them in bone marrow failure syndromes. So just a little bit of a quick overview of the bone marrow. We probably all know what it is, but it's our blood cell factory, and it's responsible for making all the different types of blood cells in our bodies that we need on a daily basis. This includes our red blood cells, which transport oxygen around our body, blood cells, which help us fight off infections, and platelets, which help us to repair tissues and clot. And so all of these cells arise from this hematopoietic stem cell, or HSC, which is what I'll be talking a little bit about tonight. So most of us know that bone marrow failure syndromes are rare conditions, and this is where the marrow can't make enough healthy blood cells. So it could be a specific type of blood cell or many different types of blood cells. These can be either inherited or acquired during lifetime. And for all different types of bone marrow failure syndromes, there are lots of different treatment options. So it really does depend on the type of bone marrow failure syndrome, but it can include things like blood transfusions, therapies such as steroids, immunosuppressive agents, chelating therapies, and other types of drugs, and also bone marrow transplants. Now, I certainly don't need to tell this audience about how intensive these therapies can be, how invasive that they can be, and that sometimes they unfortunately don't work. So looking specifically at inherited bone marrow failure syndromes, there are key proteins that might be missing, they might be faulty, or they might be made in the wrong amounts. So just to give you some context around this, in terms of Fanconi anemia, the proteins needed for DNA repair might not be working properly. In the case of Diamond-Blackfan anemia syndrome, proteins which are needed for ribosomes, which are might be missing. And in the case of dyskeratosis congenita, proteins for maintaining chromosome ends, which we call telomeres, might be impaired. When we think about acquired bone marrow failure syndromes, however, the causes may not be fully understood. So there might be an autoimmune component to this, where the body's immune system attacks its own marrow. It could be due to exposure to chemicals, drugs, or radiation. It could be through infection, but there are also lots of unknown causes that we're still trying to investigate. So circling back to what Eduardo talked about in terms of what mRNA therapies are, they really can provide cells with a temporary replacement instruction set. So think about it as a photocopy of a recipe that we can deliver directly to the cells that we need it to go to. So we might be able to make the missing protein directly. We might be able to boost the production of healthy versions of the faulty protein in the cell. We might be able to deliver enzymes to help repair DNA or maintain telomere length. We can also deliver the construction workers into these cells to help edit the genome to correct the gene mutation that might be causing the functional impact in those cells. We can also think about mRNA therapies a little bit more broadly in terms of how they can support the bone marrow compartment. So we might be able to identify therapies or mRNA therapies that help stem cells survive and repair themselves, blood cell production. And for those that have to undergo bone marrow transplant, we might be able to use mRNA therapies to help support the recovery of the bone marrow after chemotherapy, so after conditioning or after engraftment, and to help support that engraftment process. So hopefully and collectively, you can see that there's a huge opportunity here for us to be able to adapt specific treatments for individuals with bone marrow failure disorders, which really depends specifically on their requirements. So if you asked me 2+ years ago about whether or not this would be feasible in bone marrow failure syndromes, I probably would have said it's a bit of science fiction. But 2 years ago, actually, a couple of weeks ago, this paper came out in Science, which is a highly prestigious scientific journal. And although it's not a bone marrow failure syndrome, it's actually a blood disorder called sickle cell anemia. And so these scientists in this paper, what they did was they took this sickle cell anemia preclinical mouse model. So it's a genetic blood disorder which impacts primarily in the red blood cells. And so what these scientists were able to do was to deliver the mRNA payload directly into the hematopoietic stem cells, or the HSCs, in the bone marrow in these mice via one injection. And so this injection enabled the mRNA to go to the HSC compartment to start functioning properly again, and start to produce healthy red blood cells. So this is really, really interesting. And so how then could we adapt this to humans? And this is an area that's really being actively explored at the moment. There's a lot of work going on in what we call formulation science. So how we can deliver these messenger RNAs. So just to give you some context here, mRNAs themselves, we can't actually inject them directly into the bloodstream. We need to coat them with a fat. We call it lipid. And so the lipid-coated mRNA, we call a lipid nanoparticle. And those lipid nanoparticles, we can actually coat with different molecular agents. So we can actually add keys onto the surface of these lipid nanoparticles, which will unlock when they come into contact with the hematopoietic stem cell. So this particular key we call CD117. And so when these lipid nanoparticles are injected into the bloodstream, they inevitably make their way to the bone marrow. And so once they're in the bone marrow, they're able to reach the HSCs, and through this key and this lock on the cell surface of these HSCs, deliver the messenger RNA payload directly into these cells. This is, as I said, an area which is rapidly evolving. This is one way to do it. There are multiple ways that are being investigated at the moment, but this is just one way we can do this. So thinking back to my lab, so through collaboration with Eduardo, my PhD student Olivia Delfino will be using computational design and AI to develop new mRNA as potential therapies for the treatment area of interest. But I also have a bunch of preclinical models in my lab to be able to assess the function of these messenger RNAs. And so what we're going to do in the lab is to see whether or not these designs, which Olivia and Eduardo come up with, can restore protein function and improve blood cell production. And so this piece of work that we will undertake will be really vital as a first step for us to understand whether or not these therapies might have therapeutic benefit in across bone marrow failure syndromes. We can then look to extend those out, but also whether or not these therapies can be safely tested in patients. So, of course, there are some potential challenges for mRNA therapies, and these are specifically related to bone marrow failure syndromes. In that it's still difficult to get messenger RNA efficiently into the hematopoietic stem cell. So I showed a quite nice diagram of how this can actually happen, but in reality, the messenger RNA doesn't get into these cells super efficiently. So there's things that we're going to be doing to help try and enable this process to become more efficient and also ensure that we're not having any unwanted side effects. Messenger RNAs inherently are very unstable, so they don't last in the cell for very long. And so what we wanted to do is to figure out how we can extend their longevity inside a cell, but also what we can do is test to see whether or not we can deliver these repeatedly. And we also want to understand then whether or not this will be effective and safe, of course, and convenient for an individual that might need to take these therapies. As Eduardo talked about, in terms of the immune response, we can design mRNAs to evade the immune system, but it's not 100% perfect yet. So there might be some things that we need to iron out in that regard, but we can do that through testing and also computational analysis. In terms of their complexity, so some bone marrow failure syndromes involve many different genes, or they might have a complicated molecular pathway. So one mRNA itself might not be enough for a therapy. But what we can think about doing is looking at combining different mRNAs, having 2, 3, 4 different types, which might be able to support a specific therapeutic regime. And then finally, clinical testing. So all of the work that we do in the drug discovery space, we need to ensure that it's safe and it's effective, and that we've done extensive work to take it into clinical trials to then be able to establish whether or not it can be moved into use in patients. So just to quickly summarise, our goal through the NTRP and also through this project that we're running is to identify targeted therapies that might provide safer and more effective options for people with bone marrow failure syndromes. We're starting off with Diamond-Blackfan syndrome, but the knowledge that we'll gain from this project could be expanded across other bone marrow failure patients in the future. How are we going to do this? Well, there's some excellent researchers under the MRV umbrella in Australia and also across the world in different types of bone marrow failure syndromes. So working in collaboration, we can help to tailor treatments to each condition. And finally, before I hand over to Brett, I just want to say, as a drug discovery scientist, your voice really matters in this process. Hearing your experiences, hearing your hopes, hearing your concerns, these are all really important parts of what we do to try and incorporate all of this feedback into developing safe and effective therapies that you're going to have confidence in. So with that, I'm going to hand over to my colleague, Brett, now, who will talk more about that.
Thanks so much, Amy. I will just quickly introduce our next speaker, Associate Professor Brett Schultz. So Brett is a critical health psychologist at the Australian National University School of Medicine and Psychology. And Brett will be speaking with us this evening about consumer engagement. And then the survey, which, as Amy's just alluded to, is a wonderful opportunity for you to share your voice, your experience to really help inform this exciting work that's taking place here. All right, over to you. Thank you so much, Brett.
Thank you so much, Amy, and thanks also, Amy, for Amy George, for giving me a really great segue and leaving that presentation on talking about how your voice matters, because that's essentially what I want to speak about over the next couple of minutes. So I'm a little bit different to some of the other people in our team. I don't have mRNA expertise and I don't come from a biomedical background. My research is really interested in how we can make sure that people with lived experience of various conditions, including bone marrow failure syndromes, and other conditions as well, to make sure that their lived experience is brought into the research that we're producing and is part of everything that we do as health researchers. Both Eduardo and Amy have talked a little bit about the mRNA platform that our broader project is working on. And I guess I'm interested in making sure that that platform truly collaborates with people with lived experience and with community members. So over the next 2 slides, I'm just going to talk about 2 ways in which we do try to build in that collaboration and to talk a little bit about how there may be opportunities to continue to collaborate with us on the mRNA platform. So as I said, there's 2 things I'm going to talk about, and the first is the kinds of outreach that we'd like to do. I guess, this picture here is in one of our labs. These are members of our team, and a lot of what we do in this project is done within the lab, which is done in, a very specific building on campus. But we want to try and make sure that we are doing outreach and we're bringing the science that we're producing out to communities to discuss with them, to find out their perspectives. I guess, tonight's event is one example of this, but there's other ways that we want to continue to, as our platform develops, as our mRNA therapeutics develop, and as we continue down this path, to think about other ways that we might collaborate with you all and make sure that we're getting out of our labs and speaking to people and collaborating. So to that end, one of the things that we would like to invite you to participate in is a very preliminary survey. There's a link in that QR code on the screen, but I'll also pop it in the chat after I finish sharing my screen, and the link will be sent out after the event as well. So don't feel like you have to, start it right now, although you can if you want. Essentially, a very preliminary first step that we're taking is a survey to find out about people's thoughts about mRNA therapeutics, to find out whether what we've discussed with you tonight has changed how you felt about mRNA therapeutics, what other things you'd like to know about, and I guess we'll watch that over time, and as we continue to progress our mRNA research, we'll come back to that and hopefully continue to develop the ways that we outreach and the ways that we collaborate with or reach out to people with lived experience and consumers. And then the second part of our approach of consumer engagement with this mRNA platform is to also work on inReach. So I'm also really interested in how we can make sure that people with lived experience of bone marrow failure syndromes, but also of other conditions that our research team will be looking at, can be part of setting research agendas, of telling us about their research priorities, of making sure that the lived experience perspectives that you all have actually inform the science that we produce. I guess as Amy George left on that last slide, your perspective absolutely matters, because we want to make sure that we're producing therapeutics that are safe for you, that are going to actually be fit for your needs and fit for use. And so I guess as we're still in the early stages of this now, and this is something that we'd like to continue to socialize with you as we develop, and we'd love to make sure that we're keeping people updated as this platform takes shape. But I'm really keen to chat with you all at some stage, or answer any questions you might have tonight, or to keep feeding information back to Maddie's vision and other important partners about how we can actually make sure that the lived experiential expertise that you all bring informs the science that we do. I think I'll leave my session there, but happy to answer any questions about what that might look like later on this evening.
Thank you so much, Brett. And It really is such a wonderful opportunity for Maddie's Vision and our patient and family community to work with research partners like ANU, and share those experiences to inform that work. So we're really excited to be working with the team and making sure you have the opportunity to participate. I now have the pleasure of introducing Dr. Lucy Fox. So Lucy is a haematologist with dual qualifications in medicine and pathology. She leads the genomic haematology clinic at Peter MacCallum Cancer Centre and Royal Melbourne Hospital, caring for patients with bone marrow failure syndromes and inherited blood cancers. Lucy is also a clinical research fellow at Monash University, focusing on the aplastic anemia registry and the Australian Marrow Failure Biobank. So Lucy will be presenting on a clinical perspective, so current treatments for bone marrow failure syndromes and where mRNA therapies could take us. Thanks so much, Lucy.
Thanks for that, Amy. Good evening, everyone. It's a pleasure to be with you all tonight and talk about this really exciting aspect of novel therapies, really, for our patients with bone marrow failure syndromes. So one of the things that I was going to touch on was, where we are at the moment, and Amy's already done this. And one of the challenging things about presenting one of these slides is in the bone marrow failure syndromes, it's such a diverse group of diseases that it's really hard to sort of give a one size fits all approach, I guess, to treatment. But just to acknowledge that there are some principles of treatment, such as supportive care, where we give blood and platelet transfusions and supporting people from getting infections. But we know these things can be hugely burdensome for patients. We know that in some bone marrow failure syndromes, growth factors or bone marrow stimulating agents can work. They don't work in all of the bone marrow failure syndromes. But again, these things can be really helpful, but often don't provide the full answer. We know that in people who have a significant immune component to their bone marrow failure, like immunoplastic anemia, we know that we can suppress their immune system with very strong immunosuppressant drugs such as ATG, cyclosporin, but these again can come with a whole host of complications and challenges. And also we have things, so in the steroid family, if you like, we have androgens such as danazol, which can be helpful in things like the telomere biology disorders to try to stabilize or even elongate the telomeres that are short in telomere biology disorders, but they won't work for everyone. And also in the steroid family, we know that some patients with Diamond-Blackfan anemia can respond to prednisolone. That's great. It's a cheap and easy drug to give, but giving it to someone long term is a really hard ask. It comes with a huge number of complications, and we really ideally wouldn't have to give them long term prednisolone therapy. And I guess the unifying treatment for bone marrow failure syndromes, all people that I consider to have bone marrow failure syndromes, we have potentially in the treatment arsenal, I guess, is an allogeneic stem cell transplant, whether you've got immunoplastic anemia or a germline, inherited bone marrow failure syndromes. These are potential therapies, but again, these are not easy to deliver. We know people who go through these have a really tough time and potentially long-term side effects after them. And some people with bone marrow failure syndrome simply aren't well enough or too old, or for other, perhaps some reasons that they are unable to undergo a stem cell transplant. So I wanted to talk a little bit about how what a novel area this is for us in haematology, and I think and that's not meant to be a negative thing about RNA therapies, but it's just to say that this is something we're really only in recent years beginning to hear about. And things are moving so fast. We really hope these things are coming for our everyday use in the not too distant future. But to show you how recent all this stuff is, that we had a couple of years ago in the Blood Journal, which is one of our most valuable hematological papers. We had a series, an introduction on RNA therapeutics. And what Amy and Eduardo and the team talking about tonight is mRNA, and there's really other forms of RNA therapeutics, and so it can get really complicated, and the terminology can get really challenging to understand. But I just want to touch briefly on these three conditions that they mentioned in this blood review series. You don't need to remember the names of these conditions because they're not bone marrow failure conditions. The porphyrias, amyloidosis, haemophilia. And the reason they touched on these diseases back in nearly 2 years ago was because these at that time were the areas where we'd gotten the furthest with clinical trials and getting regulatory approval for use of them. And so, an example of this, and I'm recognizing that, tonight is about mRNA therapeutics, and this is slightly off that, is an example of porphyrias. And porphyrias are rare as hen's teeth, but for people who experience these diseases, they're terrible. Lots of day-to-day problems with things like tummy pain, and this says neurovisceral attacks. So one of the porphyrias are is when we have a problem in one of our synthesis pathways, I guess, where we have a problem with making heme. And so we get an overproduction of these things in the porphyrias, and that causes some really nasty side effects. And so what we have in the RNA therapeutic space is something called givosiran, which is it's not an mRNA, which we've already heard about. It's what's called an inhibitory RNA molecule, and that gets in there and interferes with this ALAS1 mRNA, and stops that overproduction and is thought to help those symptoms. And so that's quite an exciting thing for patients with these inherited porphyrias. And then again, I acknowledge I'm not talking about a bone marrow failure syndrome here, but this is a hugely important condition for people who experience haemophilia. Haemophilia is a terrible inherited bleeding disorder when you have a lack of one of your important blood coagulation factors, so either factor 9 here in red or factor 8. And we know these people have terrible troubles with bleeding into their joints. And anything we can do to alleviate that bleeding concern is really attractive in treatment of haemophilia. And so just to say that in these diseases, there's a couple of different RNA themed, I guess, treatments where we're looking at increasing these coagulation factors or reducing factors that might interfere with our ability to make a clot. And we are again quite relatively far down the road in the terms of haemophilia RNA therapeutics, but you can see 2 years ago, at this point, that people had these great ideas about different RNA therapeutics, but at that time, only one clinical trial and just ideas about possible future trials. And so, where in the bone marrow failure space, do we see these things as being exciting? And I knew Amy was going to touch on Diamond-Blackfan anemia. So to talk about another group of bone marrow failure diseases is the telomere biology disorders. And I appreciate this is a bit of a complicated picture, but for those people who experience telomere biology disorders, think of these as these little yellow dots on the end of our chromosomes. Where we all have these telomeres, and over our lives, they gradually shorten. They're there to protect our DNA, our genetic material that's sitting there in our chromosomes. But we know that people who are born with telomere biology disorders are really born with these critically short telomeres. And so essentially, the chromosomes just run out of steam. And one of the manifestations for that is profound bone marrow failure or a blood cancer at a relatively young age. And the telomere biology disorders can be caused by mutations in a number of genes, and you can see the names of these genes here, like TERT, TERK, ARTEL1, and there's a thought that that we could use mRNA therapies to really help people with all kinds of telomere biology disorders. What we hope is that for example, TERT is one of the most important components of telomere maintenance. TERT is an enzyme which actually works to elongate our telomeres. And so if you have a mutation in TERT, then you may well have a telomere biology disorder. But what we hope is that even if you've got a mutation in RTL1 or TERT, that if we can provide mRNA therapy of TERT mRNA, what we hope is that can actually help that person with short telomeres from any one of these reasons actually elongate their telomeres and have less of those telomere biology disorder manifestations. And so we heard from Amy about the idea about how to deliver these therapies to the right tissue is challenging, and that's something that obviously will be looked at in all these projects. But we obviously think about the bone marrow failure space, trying to help people with those low blood counts. But a person with a telomere biology disorder may present with just lung disease, and that's the main manifestation of their trouble. And so, there's also people that we collaborate with are wondering, well, how can we deliver TERT mRNA directly to the lungs, to help with that problem there? Could that be helpful to the lung tissue? So there's a lot of really interested people working on this, and Professor Ritchie, who might be able to join us, is working closely with an international group to try to bring clinical trials to Melbourne. And again, this is, when we go off to talk to conferences in the States or in Europe, these are the kind of talks that we sit there and listen to, from a group like this on the screen that we're really interested in. And they're very sciencey. They're often talking about mice, but they really interest us and excite us about, could these therapies be a treatment for something like Fanconi anemia? And I won't dwell on this, but only say that this group took mice that they had raised with Fanconi anemia, and and you can see here, in this case they delivered the RNA therapy directly to the sort of bone marrow tissue, and you can see that when they were able to deliver an mRNA therapy to a mouse that had Fanconi anemia, this profound empty bone marrow that they would normally see with age is reduced. And while this is not a completely normal a normocellular mouse marrow, it's still much better than an empty picture down there. There's many different ways we think about how mRNA therapies could help us, whether it's replacing missing proteins, as Amy said, boosting the stem cells, or other ways we can boost the healthy stem cells that might be there to work. Could we use these therapies to just a little bit to try to help people stay safe and healthy while we're doing that couple-month workup for bone marrow transplant? Could we while we maybe can't fix every single manifestation of a inherited bone marrow failure syndrome, even if we could do something that improves, for example, the red blood cell production in a patient with Diamond-Blackfan anemia and reduce that dependency on blood transfusions, would be absolutely life-changing for our patients. It's absolutely, this is all in the realms of this thing that we call personalized or precision medicine, which we're quite obsessed with today. And this is something that I focus on every day. It's not good enough for me to hear that a patient has Diamond-Blackfan anemia. I want to understand what gene, what is the exact nature of their mutation that has led to them having that condition. And so this is my last slide. I just want to advertise our registry and our biobank, which are supported by Maddie's vision. It's these things which I can't encourage you strongly enough to participate in. And you might not be able to participate in the biobank yet, because we're just starting with our preliminary phase of recruiting sites to the biobank. But hopefully, our dream is that every single patient with a bone marrow failure, whether it's Diamond-Blackfan anemia, immunoplastic anemia, has the opportunity to contribute their de-identified clinical data to the registry, so we understand, the nature of a person's disease and their genetics, if that's relevant, but also samples, whether it's blood, bone marrow, hair, whatever a patient is willing to give us. We want to get them in the samples. So if Amy and her team turn around today and say, do you have any Diamond Blackfan patient samples in the biobank that we can use to start, working up these mRNA therapies, we'd love to be able to turn around and say, yep, we've got 10 great sample sets. So please do hassle your doctors about this, because, the more the merrier, and we really want to offer it to all Australians. So I'm going to stop there and stop sharing.
Thank you so much, Lucy. That's fantastic to hear all those possibilities. I am thrilled to invite Professor David Ritchie to facilitate the Q&A session this evening. So David is a haematologist and bone marrow transplant specialist at the Royal Melbourne Hospital. He's a founding board member of Maddie Riewoldt's Vision, chair of our Scientific Advisory Committee, and director of our Centre of Research Excellence in Bone Marrow Biology. So welcome, David.
Thanks, Amy. Can you hear me okay?
Yes, great.
I'm not sure if I'm sharing to all of the poor victims, participants on the call, my video, but if you can see me, I'm sorry about that. If you can't, I'm also sorry about that.
We could see your thumb then, David. Now
okay.
We've got you.
All right, great. Thank you. So yes, a little bit of a rush this evening with a few other responsibilities. So sorry to join a few minutes late, but I've managed to hear the majority of the presentations, and an excellent job done by everybody. I know that there was some questions put to through Jaya and Cherie in preparation for today's presentation and information, but does anybody have any questions from the participants for our expert speakers? If so, I think the format is raise your hand or yell out, take yourself off mute and ask any questions. Just as you're gathering your thoughts about that, I might get Jaya just to have a look at the submitted questions and perhaps select a couple. But I'm going to actually ask Amy George a question. Amy, so two things have come out from a combination of your and Lucy's presentations. Number one, the hugely exciting potential for essentially protein replacement to make up for a deficit in hematopoiesis specifically. But as Lucy's outlined in her presentation, some bone marrow failure syndromes have impacts on other organ systems. We won't list those necessarily, but Amy, where do you see the ability to deliver mRNA-based therapies to other organ systems? For example, epithelial surfaces for patients with telomere biology disorders, or Fanconi anemia because of the increased risk of secondary malignancy, hepatic transfer, or pulmonary, as Lucy touched on her presentation. So do you think that targeting element that you've got for bone marrow stem cells will be able to be duplicated for other tissues?
Thanks, David. Absolutely. Yes. There's work going on for all sorts of different conditions at the moment. So that thinking back to that fat molecule that we coat the messenger RNA in, if we can put different elements into that fat molecule, that will target it to specific tissues. So yeah, certainly could go to the lung, could go to the liver, could go anywhere really. So that work is certainly being done, and it's a science in itself, these formulation scientists that are developing all of these different types of lipids, so different types of fats that will help us to take them to specific cell types in the body. Yeah, so it's very exciting. Yeah.
And the second sort of related question to that, and again, it was touched on some of the examples that you and Lucy gave, is about one of the safety mechanisms of mRNA-based technologies, of course, as you've highlighted, is the fact that they are temporary messengers. Literally, they don't have a long persistence in the tissues. So what do you feel the requirements will be of these technologies for repeated dosing, and how will you measure the potential that the therapeutic effect of dose number 1 might be waning and need for a second dose?
Yeah, that's a really good question as well. So we're not really sure at the moment. This is work that we're going to have to do in our lab. So we're really going to have to look at repeated dosing. We have mouse models of DBA in our lab. So that's something that we'll certainly, once we have some messenger RNAs, we'll definitely be doing for sure. Yeah, absolutely. I'm just trying to think back to the first part of your question, which was around what we can deliver in their longevity. I guess, you know, we can obviously we can deliver a replacement messenger RNA, so that may not last very long. But what we can do through computation is we can actually design different elements as part of that messenger RNA that help with that longevity. So that's one thing that we're certainly going to be doing with our DBA project. Will be changing up the sequence of the messenger RNA so that we can make it more stable. And the other thing that we can do as well is that we don't just have to deliver the copy of the faulty gene. We can actually deliver construction workers to the cell to help potentially gene edit in situ. So that paper that I showed before was one example of that, where they delivered CRISPR molecules to the HSCs to correct the gene defect. Of course, there are some questions around the safety of that. But these are certainly things that are being investigated at the moment.
A couple of questions on the chat, and I'll read them out. So thanks to Leah and to Karen for submitting, and Amy's also popped one in from a pre-submitted question. We'll get to that in a second. So I think you touched on this in your presentation, Amy, about the possibility of targeting different tissues in different ways with different preparations. You see it in the chat yourself. Do you think it will be feasible in the future to deliver multiple formats of mRNA therapies, one that might have an advantage in the marrow, whereas another may have advantage in another tissue, such as the lung or liver? And do you see those essentially being compound therapies?
Yeah, that's a really good question as well. We're not really sure about this, but there is so much potential. I mean, if we think about, for example, trying to deliver to the lung, we might be able to formulate particles to inhale, and then if we want to target the bone marrow, formulate that and inject through an injection, you can deliver straight to the HSC. So I think there is a potential, but again, because this area is so new, we're not really sure yet. So these are things that we certainly want to examine and understand, you know, what's the level of tolerance that we have to being able to deliver mRNA in different modalities in the same individual at the same time. It's going to be a really important question for us as scientists to answer that.
In the chat, an excellent question is, you know, so there's excellent examples of models in vitro and in vivo, and Lucy shared some of those as well. And I might get an answer from both you, Amy, and from Lucy. Is, you know, what does the timeline look like? How soon do you think there may be preparations that can move from the research environment into a clinically deliverable, even if that's within a clinical trial research context? What sort of timeline do you think is the field moving at? Amy first, and then Lucy.
Sure. And yeah, Lucy, please jump in if you have anything to add to this. But I would probably say I would probably encourage everyone to think back to COVID times, where we didn't have a vaccine for COVID-19, and then within, I think, the space of 6 months, there was a messenger RNA therapy that was ready to go. The wonderful thing about these mRNA therapies are they super fast, super adaptable. So, you know, we're not thinking about, as a drug discovery scientist, when I'm developing a small molecule, it might take us 10 plus years for us to be able to take it into the clinic, whereas these would be in the space of 6 to 12 months potentially. But as I said, we need to do a lot of work around the safety profile of these. We need to get that preclinical data so that we can actually ensure that they are safe and they are applicable and that they're going to be effective, right? We don't want to deliver things that are not going to be effective to patients. So, yeah.
And Lucy, what do you feel the timelines that we're looking at are going to be? You have, you know, patients in the clinic who may be very anxious to have access to novel therapies. What would you tell them if they asked you?
Yeah, so look, it's obviously difficult to be very precise, but I think it's reasonable to hope that we would have clinical trials at least, you know, in within the next 3 to 5 years. You know, I think that's really reasonable. Hopefully even earlier. We know we have collaborators around the world who are encouraging us to apply for, you know, grants so we can implement clinical trials here. So some things are a little bit funding based, but we know that we have collaborators who you know, would be ready, you know, to start up a clinical trial within the next year or so. You know, acknowledging all the sort of the challenging infrastructure and everything. And then I guess you know, one of the other challenges we have, you know, with these diseases, with these conditions, you know, and I touched on in my talk, sometimes it takes us, you know, a year to make, or longer, or never, to make a really good, accurate diagnosis. I'm sorry if you can hear my dog barking. Yeah, so you know, you know, the sooner we can get an accurate pinpoint genetic diagnosis for people who have an inherited thing, you know, the sooner we can you know, get them in the registry and then hopefully into the biobank, so we can really quickly turn around and say, you know, to our colleagues overseas, to our collaborators overseas, you know, we've got 5 patients who, you know, absolutely fit the bill. And so, yeah, I think I don't think, you know, 3 to 5 years as being unrealistic.
Great. So there was a second part, and I really would like to sort of, I suppose, bring in the view here of sort of implementation scientists and the voice of the consumer when designing research. And so, because I can't see all the recipients, I can't remember who our third speaker was, but I'd really like to bring him into this conversation at the moment. So maybe first from Amy, Angela asks, so we're really talking here about potentially a longish term therapy for a gene that is missing, and here we're talking about mRNA therapies, but it's not likely to be a permanent cure. It may require multiple treatments. Whereas I think from what you were saying and using the analogy of the sort of the construction workers, the gene editing is likely to result in a permanent cure, at least in the tissue that was successfully gene edited. So do you feel that there might be some therapies that are once off and essentially permanent, and others that might be repeatedly therapeutic? And where do you think the optimal timing of the introduction of those therapies might be? And does it put pressure on the clinicians out there to identify patients as young as they possibly can be in order to benefit from these therapies? So first of all, are we talking about a corrective therapy that's not permanent, or are there potentially permanent therapies? And ultimately, where do you see the best benefit from an age of the recipient? And then I'm going to go to, if somebody can remind me of his name, is it Brett or Brent?
It is Brett, yes.
Brett, thank you. And I might just ask Brett, what structures do you think would help from people with lived experience, from patients, their families, from future consumers of these therapies? How would their voices best inform those ideas about what one therapy over a different therapy and the optimal timing of those. So I know it's a complex question, but we're going to start with Amy first regarding the permanency of the treatment, and then we're going to ask Lucy again about the timing of the intervention, and then we're going to ask Brett about the how patients and their families can help inform those decisions regarding the direction of the research. Amy, you're first.
All right. Okay. All right. So I would say that we have, going back to my slides, is that we have this opportunity to explore a number of different ways. So I think initially what we're going to try in our lab is, can we just deliver that recipe to the cell, and how long is that cell going to have that recipe in it, and how long is that going to be effective? Because if we're just delivering in a messenger RNA by itself, the safety profile is reasonably good. It's going to degrade. It's not going to change the genome of that cell or the DNA of that cell. But I think there's opportunity for us to be able to explore other modalities. So there's things that I've not talked about tonight called self-amplifying messenger RNAs. So they're like a mRNA on with another factor attached to it, which helps it to amplify in the cell. So it doesn't change the DNA in the cell, but it just sits in part of the cell near where the proteins are made, and it's sustained. So we're going to test that out. But of course, going back to the gene editing, I think that's something we also want to explore. We want to see whether or not we can correct that and make sure that the components that have done the correction have exited those cells, and what effects that has on that. So I think I'm writing plenty of NHMRC grants at the moment around this, because we like to explore all sorts of different avenues here. In terms of the question around pediatric patients, and I'm not quite sure how to answer that one. I'd probably defer maybe to thinking about the COVID-19 vaccine, and I think, and please correct me if I'm wrong, clinicians, I think it was given to patients 2 and above, maybe, or 5 and above, I'm not sure. Or there was some conjecture, I know, at the time. I can't remember what the age was, but that's something certainly that we would need to have discussions about and certainly keep in mind going forward.
And yeah, thank you. Agree. Really good that you gave me that COVID idea as a segue there, Amy, as well, because I draw on that in answer to the second part of the question. So I think safety, obviously, for patients is paramount, and once we felt that there was safe therapeutics and there was access to that, I would like to see more co-design into what clinical trials might look like. So we're not alone in having a co-design approach, but one of the strengths of the co-design approach of the mRNA platform is that we are interested in looking at how we can make sure that people with lived experience are part of designing all the things we do. So when we get to a trial point in this kind of research, I would envision that one of the really important steps would be having a range of people with lived experience of all different kinds of conditions coming together to talk about what that would look like and how to make sure that they all felt safe to be part of a clinical trial of this kind of thing. I guess if you're anything like me, the first time I didn't come from a medical background, I'm a psychologist by training, and the first time I ever heard of mRNA was in 2020, and I think bringing people along on the journey was something that had to be done really quickly in COVID, but we actually have a little bit more time now to make sure that people with lived experience are part of designing these things early on and throughout the process, and I'd love to see that continue, and I think that's part of what we want to do with our platform.
If you don't mind, Brett, might just tease out that idea a little bit, because we have a lot of patients and their family members and people who are really interested in supporting novel research, and I know you've made reference to a questionnaire that's part of a bigger project that Amy's run. What future forms or future mechanisms might people with lived experience, patients and their families, how might they actively engage in that process? So I think you've been very clear about how important those voices are engagement.
Yeah, thank you. It got a little bit choppy there, but I think the question was, what are some of the specific mechanisms for engagement? Yeah.
Yes.
Yeah. Um, yes, absolutely. I mean, I guess as far as our projects are going, we're still in the piloting phase. So we're early on. I mean, we do want to embed co-design in everything that we do, and we actually don't yet have the mechanisms for how that will look like at that when we're at that ready for trial stage. I guess we're at the preliminary stage where this survey, for example, is one of the very first steps just to look at very preliminary ideas and isn't yet at the co-design phase where we're actually bringing people with lived experience in. But we want to, and I guess we see that as something that will happen at all stages of our platform, even in the governance level. So I guess we'll continue to be liaising with organizations like Maddie's Vision, and some of our other key partners, and opportunities as they become available will be sort of disseminated through those partners. So for people who are interested, please, we will continue to engage some of those partners, but would love to hear from you when those opportunities arise.
Okay, I'll take a cue from Amy Coote about if our time is up, but there was a pre-submitted question, and might answer that question because it was a question where, for various reasons, but certainly can occur in bone marrow failure syndromes of a range of different types. And really, I suppose, just to channel Amy George's views, is that really these technologies and these therapies are about providing a new set of encouraging directions to the bone marrow to improve its productivity. And so if there is an identified absence of something in the bone marrow that leads to neutropenia, then correcting that with these types of therapies may very well correct that particular low white blood cell count problem. Amy Coote, how are we going time-wise? I think my clock, we're probably about done.
Yes, David, we have gone over time, but all for very good reason. So we will need to wrap up the session, but for our patients and families on the call, you know our number, you can reach out, and if there's any additional follow-up that we can do on your behalf, we can absolutely contact the ANU team and their bright minds and get some further answers for you. But I would like to, thank everyone for joining us tonight for this educational session. We really hope it's been informative and very helpful. But again, just the importance of having our patients and families share their experiences and share their voice to inform everything we're doing at Maddie's Vision, but also that work of our patients and families. And as Brett alluded to, it's early days for this particular project, but we look forward to working with the team at ANU to identify other opportunities that we can create over the coming years to bring you in to inform this exciting work. So I would like to thank our guest speakers, Professor Eduardo Iraz, Associate Professor Amy George, Associate Professor Brett Schultz, Professor David Ritchie, and Dr. Lucy Fox. We're very grateful for your time and your amazing expertise, and feel just really privileged that we have this incredible network of amazing people that we can bring together to share these insights. So we will be sending a follow-up email. So the first will be from Maddie's Vision, which is designed with a survey. So that will be designed to help us improve future educational sessions. So please do share your feedback on the session, the content, and how much you've learnt from this evening's session. And the second, which Brett did discuss, will be coming from ANU. So that's really aiming to understand your thoughts and attitudes and awareness around these mRNA therapies. So we really appreciate the time you take to complete those, but it really does help shape our work at Maddie's Vision and that of our research partners. So tonight was our third patient and family educational session for 2025. It's hard to believe that we're already coming towards the end of August, but there will be a final fourth session coming up in the coming months. So please, could you keep an eye out for that next session, and we look forward to bringing our wonderful community together for that again. I think we're going to try and send the link in the chat for the survey, but it will be coming in the follow-up as well. So thank you again for your time this evening, to the wonderful team at ANU, to Lucy and David, and we look forward to keeping you updated as this exciting work progresses. Have a lovely night, everyone. Thank you.